My project was the other half of Jennifer to benchmark the language model-based spatial foundation models. And to be more specific, I benchmarked the zero-shot abilities of scGPT Spatial and Nicheformer. Okay. Yeah, just a little bit background. Why are we building, why do we need a spatial foundation model? I think it's because spatial foundation model can give us a unified, generalized, and interpretable representation of our spatial data. And based on that representation, we can do a lot of downstream tasks like understanding the tissue microenvironment and maybe predict drug response and so on. And there are two major challenges to building a spatial foundation model, which first I think is the data heterogeneity. Because we use different sequencing protocols, the data may be a lot different from each other. For example, if you look at Visium, it's a spot level scale, but it can give you a whole transcriptome. But instead, Xenium and Cosmics can only give you several hundred or thousand genes, but it is a single cell level. So, how to resolve the problem of data heterogeneity will be a central question that this spatial foundation models should answer. And the second challenge will be how to incorporate spatial information into the foundation model. And for my benchmark, why did I focus on zero-shot ability? I think the promise of a so-called foundation model is its ability to serve as a base for many downstream tasks without further fine-tuning. So I tested their zero-shot ability specifically. And in the interest of time, I'm not gonna go through each of the method but rather focus on which aspect they are different from each other. So scGPT Spatial is trained on Merfish, Xenium, Visium, and Visium HD. But Nicheformer is trained on Merfish, Xenium, and single-cell RNA-seq, and also Cosmics ISS. Um, for species, scGPT Spatial is trained on only human data, and Nicheformer is trained on human and mouse data. And the dataset size for scGPT Spatial is about 30 million cells or spots, and for Nicheformer is about 110 million cells. And scGPT Spatial is pre-trained from scGPT, which is a single cell foundation model that the authors published. And Nicheformer is totally trained from scratch. And scGPT Spatial is trained in an autoregressive manner, which means it uses next token prediction when it is pre-training. And the Nicheformer use mask language modeling. And for the spatial information, scGPT Spatial use a learned spatial embedding. It used a very special technique where they sampled some of the neighboring spots or cells and try to predict the central spots or cells. In this way, their model learned some spatial information. And for Nicheformer, it didn't incorporate any spatial information during pre-training. I was a little bit surprised, which this means that they take the spatial data exactly the same as single-cell RNA-seq data, which is pretty odd, but they did use some of the spatial information during their downstream task. And for the downstream task tested in their paper, scGPT Spatial tested multi-slide integration and cell type deconvolution and also gene expression imputation. While Nicheformer tested for cell type niche and region prediction and also neighborhood density composition prediction. And so for my benchmark, I got some private data from Xiaohan, and it is a paired Cosmics and RNA-seq data, but only a small portion, so it's like very manageable. And for the data pre-processing for scGPT Spatial, I looked very carefully in their paper. So they first do a slide level normalization to normalize the gene expression across the whole slide, and then a corpus level normalization, which means they normalize each gene's expression across all samples. But for this part, because they didn't offer the mean and standard deviation of their corpus, so I wasn't able to do this part, which might have some effect on the results. And for the Nicheformer, they use the ensemble gene ID. So you have to convert your data to ensemble gene ID and then normalize to 10,000 counts per cell. And then they normalize expression by technology specific mean expression vector, which means they give a mean and standard deviation for each sequencing technology. Here I use the Cosmics and their single cell RNA-seq. And also they rank the genes from the highest express to the low express. Yeah, that's for the data processing, pre-processing. And so using their pre-trained weights, I was able to get the embeddings for each cell for the cosmic data. And so I listed all the results here. The first one is where I do the UMAP only based on the expression, gene expression. You can see that this is a pretty, the clusters are pretty well defined. Um, but when I use their embeddings, it's like a giant blob of everything mixed together. But however, comparing the scGPT spatial result to the Nicheformer result, I can see that scGPT spatial is slightly better because the mesenchymal stroma cell and also the endothelial cell and the lymphoid cell and also the epithelial cell are somewhat clustered together. But for the Nicheformer, you can only see the like maybe the mesenchymal stroma cell are clustered together and the other cell types are just a giant blob. So this I was pretty suspective of this result. So I think I tried different normalization, but it didn't help. So I go on for the single nuclear RNA data. This one looks pretty normal. You can see that the original expression UMAP cannot like separate the cell cluster from each other, but for the scGPT spatial, for each cell type, you can see that they're like clearly clustered with each other. And also for the Nicheformer, I have like similar results. And next, I try to do multimodality integration, which I realized it's not gonna happen. So before the integration, it was like a single nuclear RNA one cluster and spatial one cluster. But after the integration, I don't think scGPT spatial and or Nicheformer can do this job because it's just one is just worse than the other. But I did manage to look at some spatial cluster on the Cosmics data. So took the embeddings that scGPT spatial and Nicheformer output, and I did the latent cluster using the same parameter on all the cells. And when I visualize the latent cluster using different color on the these two FOVs, I can see that for the scGPT spatial, there are some signs of like epithelial cells being clustered together by latent cluster because you can see some cells here. And also maybe the mesenchymal stroma cell are like clustered together. But for the Nicheformer, I don't think I see any pattern from their results. So also I calculated their ARI score, which is the which is a metric that evaluate the performance of your clustering. And I compare the scGPT spatial latent cluster to the cell type cluster. And scGPT spatial did a slightly better job, but it's not as good as I expected. So yeah, that's pretty much all I got. So I tested the zero-shot ability of these two foundation models, and scGPT Spatial performed like slightly better in zero-shot cell type clustering on Cosmics data, and they both did a pretty good job on the single nuclear RNA-seq data. But both of them cannot do multi-modality integration properly, and I think scGPT Spatial produces a more interpretable spatial cluster than Nicheformer. And of course, the limitation of this benchmark is that the data set is pretty small and very only incorporate two modalities. So the results are like very limited to this kind of these two modalities. And also the lack of quantitative evaluation. So a lot of UMAP, I just eyeball it. And yeah, that's pretty much it.